Hey Sam, thank you for meeting me here.
Hey Bree, it is no problem. What did you want to talk about?
I've been studying for my cell and molecular test, but I'm confused about a few topics and was wondering if you would go over them with me.
Sure, what topics did you have in mind to start?
Can we talk about chromatin compaction from protein-free DNA to the mitotic chromosome, and the roles of HDACs and HATs in DNA accessibility?
Of course. So, to start you have to know that the chromosomes are packaged so that they already fit easily into the cell's nucleus, and they are organized so that they can be given out to daughter cells during mitosis.
And they are organized so they do not get tangled up and can be easily unpackaged?
Yes.
I know that a chromatin is any packaged DNA from the nucleosomes to mitotic chromosomes, but what do they do?
Well, when chromatin is regulated, the cells can regulate the access to DNA and achieve localized unwrapping of the sequences that will be needed.
That makes sense. What else do you know?
So the cells use a lot of ways to get the DNA sequences that are sequestered in the chromatin using their own set of proteins. The chromatin remodeling complexes uses energy from ATP hydrolysis to move the nucleosomes out of the way easily. This physically forces the DNA to unwrap from the nucleosome and thread adjacent DNA to the nucleosome. These complexes rearrange the nucleosome arrays by spacing the nucleosomes out and then exposes the underlying DNA.
It is that the remodeling complexes are inactivated during mitosis, and that is why DNA stays compacted. That is the SWI-SNF complex that is in yeast. That is this complex, if I'm not wrong.
Yes. Perfect. And the other way to access DNA using chromatin is the histone tails that come out of the nucleosome's core. The tails have amino acids and lysine, and the lysine carries a positive charge on the side chain, and that reacts with the negative charge that DNA has, so the lysine serves as a lock. Though they can be acetylated and that neutralizes their positive charge. But decetylation can restore the positive charge, and tighten up the nucleosome back.
So the negative and positive charges act as a lock and lock the nucleosome into place? Got it.
So the histone acetyltransferases acetylate histones, which loosens the nucleosomes and makes DNA accessible, which allows the DNA to repair, transmission and things like that. On the histone decetylation complexes, decetylate histones, which lock the nucleosomes into place and inhibit the access of DNA. This picture, the top shows the decetylation of the nucleosomes. You can see that they are tightly locked, versus the bottom where they are acetylated, they are more loose and spread out. Also, a little tip to help you remember what they do is by looking at the names of the two tells you the exact job that they do to the histones.
I never thought of it like that. I just thought they had silly names.
In a lot of cases, the name will tell you what the job of it is. But make sure you know the SAGA complex. It is very common in the histone acetyltransferases in yeast. The SAGA often is the one to loosen the nucleosome first, and then the SWI-SNF that you talked about above moves the newly loose nucleosome. Is there anything else you'd like to talk about? Is there anything else you'd like to talk about?
Can we talk about DNA replication? I know it is the process of making identical copies of DNA right before cell division, but I do not know much else.
So to understand DNA replication you need to understand the parts that go along with it, so I am going to start with the replication origin, which is where the DNA synthesis begins for the specific sequence. Next is the semi-conservative replication, which is where half of the original DNA serves as a template for the new strand, and the new DNA have one old and one new strand. Going on to DNA helicase is an enzyme that unwinds the double helix and separate the DNA strands for DNA replications. The DNA polymerase is an enzyme that joins the individual nucleotides to get DNA, though RNA primer is also important, and the enzyme that creates the primer is called primase. RNA primer is responsible for the RNA nucleotide sequence that is bound to a single-stranded DNA and that initiates the replication. Some of the most important things to know is that the leading strand is continuously synthesized into the replication fork, which is a Y-shaped region where the new strands grow, but the lagging strand is synthesized away from the replication fork in fragments that are called Okazaki fragments. These are put together by DNA ligase to form complete strands by forming covalent bonds. the direction is always 5' to 3'. I like this picture because it holds a lot of the information that I just talked about. You can see the helicase that starts at the far left. The orange looks like the origin of replication, where the fork starts to begin. As said before, the leading strand starts at the 5' strand, so it is continuously made with one RNA primer. The lagging strand starts at the 3' end and has the Okazaki fragments that are discontinuous and with multiple RNA primers. The bigger blue boxes in this picture is the RNA polymerase 3, which I did not talk about, but it transcribes the DNA to synthesize the 5S ribosomal RNA, which is the large subunit of the ribosome.
That is a lot of information, but I think the pictures are very helpful. Thank you for bringing them. Replication is a lot more clear to me now.
I'm glad that I could clear that up. I know on the phone you said you wanted to talk about DNA mismatch repair and the two mechanisms for repairing double-strand breaks. Do you still want to talk about that?
Yes, I know that in DNA mismatch repair that the fixes are snuck through the DNA polymerase proofreading. It fixes bases that were not paired correctly, and recognizes errors that can mess up the DNA by making a new DNA section, cutting out the mismatch and inserting, or taking the old non-mutated strand is used as a template to fix it. I know that the best way to fight somatic mutations and cancer is the mismatch repair because cancer cells inherit mutations in the genes encoding protein. I also know that in bacterial cells they have methylation to mark the correct DNA templates after they are replicated and checked, and the newly synthesized strand which is not checked is not. So when there are mismatch, the cell can fix the unmethylated strand, and once it is fixed it will match with the methylated strand, and once the new strand is known to have been fixed, it is methylated because it is trusted.
Yes, that is correct. Did you know that in base excision repair a damaged, mutant or modified base is recognized then cut out of the DNA, and that the single nucleotide gap is filled by DNA polymerase, which then adds the correct nucleotide, and the DNA ligase seals it? On another note, the nucleotide excision repair repairs the damage that causes major distortion to the structure of the DNA. It fixes most types of DNA damage. It is conserved from bacteria to humans. It does so by scanning the proteins along the DNA structure looking for distortions, and when it is found, the double-stranded DNA in the region is then separated and kept that way. The mutant strand is severed in that strand on both sides of the damage, which leaves a gap, but that gap is filled by DNA polymerase and sealed by DNA ligase. When this double strand breaks, it makes the DNA into two pieces, which could be a life-ending mutation, and that is because there is no intact strand to act as a guide, so there is ionizing radiation, which are mistakes made at the replication fork, and the free radicals cause the double-stranded breaks.
I actually have never heard of the two types of excision repairs, but it makes sense for the two types of repairs now, which are the homologous recombination and non-homologous recombination, right?
Yes. Do you know what they do?
I think so. Doesn't homologous recombination elegance, and it is thorough, but it is not always possible because it needs the DNA nearby to be extremely similar in sequence, and non-homologous recombination is quick and fast, but the cell finds it better than death. It takes the two ends of the broken pieces of the strand, then files them down and ligates them together. While the break is fixed, there are a lot of nucleotides lost in the process, and this process is usually fine unless it is done in the middle of a gene.
Awesome. There is a little more into homologous recombination. You have the first part right, and it needs to have a similar DNA sequence nearby. But the process of this is when the single strand broken end looks for a sample in the nearby sequences. When there is one that is similar, there is a branch point that is created where the two strands can cross each other, and the invading strand elongates by the work of the repair polymerase, and as the DNA is synthesized, the branch point is moved forward. When homologous recombination is done, when the nicking of the invading strands, which allows them to return to normal, the nicks are sealed with DNA ligase, and then there are the two completely intact DNAs. So this picture just shows the breaks in the homologous recombination. You can see the break in the top DNA strands. The red then goes into the blue of the very similar strand, and that is the strand invasion, and then that is when DNA synthesis occurs. The two become one at the intercross, and then the DNA ligase occurs to make them one. It's a pretty cool process if you think about it. Oh, and when this occurs, the 5' ends are what are being basically chewed away at.
Wow, I did not know that. That is so interesting that our bodies can do that. What do you know about the assembly and purpose of the pre-initiation complex?
Well, the PIC has general transcription factors that assemble along the RNA poly-2, and that is what forms the PIC, and that includes the DAB-FA, that is the order of recruitment for the RNA poly-2 for the transcription factors. So it begins with the TFIID, and it binds to the promoter, and it is made of tau-binding proteins, and TF, then the RNA poly-2, and TFIIF bind to the RFIIB, the pre-initiation complex of TFIID, TFIIA, TFIIB, and TBP bind to the promoter, and RNA poly-2 is bound to TFIIF, then binds to the complex at the promoter, and then TFII and TFIIH joins the complex. And once it is put together at the promoter, the RNA poly-2 leaves the pre-initiation complex. When the nucleotide precursor for RNA synthesis and phosphorylations of serine resides with the enzyme tails, these tails have many repeats of heptapeptide sequence. The main function of the PIC is the recruitment of the RNA polymerase, and it is absolutely necessary for transcription, and for the binding of the enzyme to the promoter region.
It is amazing that you know all of this. I cannot even think of all this. I don't think I'm going to remember this for the test.
Yes, you can. It is just a little bit of practice.
There is just so much to know.
Yes, and we can continue to talk about all the confusing topics to you.
Okay, thank you. Do you know anything about post-transcriptional mRNA processing?
This is when in bacteria the newly transcribed RNAs are immediately bound by ribosomes and proteins are made, but for eukaryotic cells, which we have nucleus and the ribosomes are outside, so that makes it so that the RNA is in the nucleus. The whole point to this is the quality control of the cell, and we inspect our RNAs prior to translation. This occurs because of compartmentalism. But the RNAs have to be processed. So there is addition to the beginning, and then after the cell has a stamp of approval, only with RNA can the additions leave the nucleus. So the enzymes that make these additions ride with the RNA during transcription on its tail.
I know that mRNA needs to have a 5' cap, and it is a weird base. A methylated guanine attached atypically is added to the 5' end of the mRNA, and this cap marks the mRNA as genuine, and the mRNAs receive a poly A tail which is hundreds of A's long at the 3' end.
Yes, that is correct. There are also 2 modification results that make RNA more stable, and it allows for exit from the nucleus and then next to be translated. This marks RNA as RNA. And for the final step, the proteins that were wanted by the 5' cap and the poly A tail interact, and this causes a good mature, genuine RNA to circularize. After all, coding and function RNAs are made through a process that is called splicing, and introns are removed and exons get stitched together. There is a complex machine that is called spliceosome that removes introns and ligates exons together, and the freed 5' end is attacked by the A in the middle, making a lariat. The lariat is a type of loop of the discarded byproducts from the splicing of RNA.
Oh, I was confused about what a lariat was every time I was studying. Awesome, thank you. Also, I know there are benefits to introns because the select pressure to keep them in alternative splicing. This is because they do not need to keep all the exons, but by splicing the non-adjacent exons together this deletes some so there can be new proteins made, and there are many different types of proteins that can be made from a single gene through this alternative splicing.
And after transcription the RNAs are checked, then capped, tailed and spliced, meaning that if they go through this process then and only then they leave the nucleus for translations, and this transport across the nuclear membrane is highly selective and regulated. And it is monitored by the nuclear pore complex. Seems like you had that topic down pretty well.
Post-transcriptional mRNA processing made a lot of sense to me. I also feel like I understand a lot of the next topic that I am going to ask you about, and that is translation.
Translation? So since it makes sense to you, why don't you tell me what you know?
Alright, so the first thing that I know is that it is basically one language to another. Nucleic acids or nucleotides. RNA is similar to DNA. It has 4 nucleotides that are held together in a 5' to 3'. So the DNA gets translated into RNA using the nucleotide sequence into the amino acids. While there are 4 nucleotides, it gets changed to 20. While one nucleotide for one amino acid does not work, even 2 nucleotides do not work. 3 are actually needed, and that the 3 nucleotides are called triples, so there are 64 codons for amino acids which allow for redundancy, and there are reading frames that start with AUG and end with a stop codon. Is there anything else?
That was great. I would just include the steps, which are: the first ribosomes assemble around RNA at the 5' end, and this happens with the repeating fashion. Then the amino acids on the tRNA form a peptide bond on the amino acid chain that is lined in the P site to the tRNA. That transfers the whole amino acid chain. There is a ribosome shifts towards one codon, while the tRNA stays base paired, which is translocations. Moves tRNA formerly in the P site goes to the E site, and then the newer incoming tRNA is now in the P site. And lastly, there are only 2 tRNAs that can be in a ribosome together, so when a new one enters the E site, the one that is in the E site gets kicked out. I'm going to show you a picture from transcription to translation. It helps me remember what translation is all about.
What is it?
I like this picture because it shows the whole RNA to mRNA to finally the polypeptide chain.
That was really helpful. Thank you. Well, there are only 2 more topics I'd like your help with, if that is all right with you.
Yeah, of course. What are the 2 topics?
I would like to talk about the combinatorial control mediator and RNA.
Okay, I will start with the combinatorial control and mediator. This is if multiple proteins are needed to turn the eukaryotic gene on or off. This leaves for the genes to respond to multiple signals, so this can depend on time, condition, and amounts. The combinational control are groups of regulatory proteins that are working together to control the expressed gene precisely, but many things regulate one gene. The mediator is just a huge protein complex that sits on the end of the RNA pol II. It relays the information. The complex gets the signals from the activators and then passes them to the RNA pol II. But it is important to know that they can send activating and repressing signals as well from upstream regulators and enhancers. They are large in size at about 20 proteins. Combinatorial control. There are transcription factors goes hand in hand with controlled expression, so in this picture it shows the 2 activators which allows for transcription, with no repressor. In the second cell there is a repressor, which means there is no transcription, so this means if there has to be different combinations for the control of transcriptions to make any given gene. And the mediator here is the complex that makes the function for the transcriptional coactivator. While this sits on the end of the RNA pol II and sends the signals from either the activators or repressors and integrates them, as I said before. But also remember that this is a committee decision, and that is how it happens for gene expression.
The combinatorial control helps with gene control and the mediator helps pass signals along. I think I understand this.
As for RNA, this is a mechanism used by cells for gene silencing and requires the formation of dsRNA. Once the dsRNA is recognized, the RNA machinery, all dsRNA and ssRNA with that same sequence is destroyed. RNA is a very potent response, and it is believed that it initially evolved as a defense against viral infection, and viruses are the primary natural source for dsRNA. RNA reacts when the dsRNA is fully base paired and approximately 21 to 23 base pairs long. The longer the dsRNA means they are cut by enzyme disser into smaller fragments. The smaller dsRNA are called small interfering RNA, and they are bound to RNA-induced silencing complex. The RISC uses one of the 2 strands that come front the RNA bound to recognize other RNA molecules with complementary sequences. Once they are, the RISC destroys the RNAs. RNA is one of the most potent regulations that is known. 50 or less of the RNAs can silence thousands of pre-existing RNAs, and that is because of amplification of RNAs by the RNA-dependent RNA polymerase. And after RISC cuts them, the 2 unstable fragments are left behind, and the RDRP appears to use one of the fragments as a template to make the dsRNA. RDRP does not need a primer. The dsRNA is then used by disser to make more RNA for the RISC. Since we lack the RDRP, we do not amplify the RNA. RNA is used a lot in a laboratory to prevent expression. It is basically a shutdown demand button, so when siRNA is expressed we achieve almost instant storage of protein synthesis. Here is the picture I used to try to understand this mechanism. So the long double-stranded RNA is cut up by the dicer, and that is when the single-stranded RNAs are made. Risk then comes into play, either blocking the translation completely because it gets stuck, or the mRNA is cleaved.
I honestly had no idea on the RNA. Thank you for explaining that for me. Thank you for explaining all of those topics to me.
Of course, anytime, Bree. I enjoyed helping you cover all these topics with you. Do you have any more questions?
No, you really helped with this. I think I am ready for this test.
Awesome, I think you are ready for this test too. I am glad I could help you. Good luck.